In science, when you want to move beyond discovery, when you've actually made a big finding and you want to turn it into reality, the type of funding we get from the federal government is not at all useful for those types of studies.
I'm trained originally as a chemist, and I've learned that there are a vast number of systems in biology where if we could just take a good look at them, we could learn all about how to modulate them.
The Blavatnik gift has really been transformative for our work.
The Blavatnik Award has allowed us to de-risk many of the components of this project to establish a solid proof of principle.
So this is really important stuff that we're doing. The Blavatnik Fund will get us to a point where we can truly commercialize.
Way too many people are touched by Alzheimer's. My father, my grandmother suffer with the disease. I'm driven to make this succeed. Although we have some medications that are FDA approved that reduce symptoms a little bit, we don't have any disease-modifying therapies for Alzheimer's. My name's Steve Strittmatter. I'm a professor in the neurology and the neuroscience department at Yale. These Blavatnik programs are really helpful to try to move these discoveries in the laboratory into drugs that we can test for their benefit in the clinic.
The great fortune just recently of having our 7th FDA approval for a new medicine coming out of Yale Research. Hi, I'm Bill Wiesler. I'm part of the Office of Cooperative Research, and I'm the director of the Blavatnik Fund for Innovation at Yale. The Blavatnik Fund is greatly accelerating our ability to fulfill our mission in the Office of Cooperative Research, which is to take the great life science innovations out of the labs and to see them turn into new medicines and new products. We're only just getting started with this fund, but already we've seen so many great impacts around campus.
I applied to develop an idea that I had about how to stimulate the innate immune system in human beings. I'm Anna Marie Pyle. I'm Sterling Professor at Yale University, and I'm also a Howard Hughes Medical Institute investigator. Based on that, I was able to design molecules that could turn on the innate immune system at will. What we would like to do now is expand on that and find new classes of molecules for antimicrobials.
Our initial goal, actually, like many academic projects, wasn't actually to make a drug. It was to test a fundamental question in biology. Hi, my name's Aaron Ring. I'm an assistant professor of immunobiology at the School of Medicine. So what we've done is we have made the type of drug that most pharmaceutical companies wouldn't typically pursue. We took a patient's own immune signaling molecule called a cytokine, and instead of accepting nature's solution, we engineered it to control how our immune system's responding to cancer, to autoimmune disease, to other aspects of our physiology.
The first award from Blavatnik really got us off the ground at the most vulnerable time for a new company. I'm Paul Turner. I'm the Elihu Professor of Ecology and Evolutionary Biology at Yale University, and I'm also a microbiology faculty member at Yale School of Medicine. Millions of people worldwide have lung ailments that make them very vulnerable to bacterial diseases. And those bacterial infections are sometimes multi-drug resistant, so there may be no drugs, literally, to help these patients. In our particular technology, it's all about getting a dose of bacteriophage, viruses that we would use in treatment, prepared properly under ideal manufacturing practices. And we've been able to do that in our laboratory for personalized medicine approaches, but to actually run a clinical trial takes a tremendous amount of resources.
One of these studies that we did with the Blavatnik gift was actually larger in scale than an entire year's worth of an R01 award. There's no way that any lab could afford to do a singular study like that. Okay? But without those types of studies, we would be in this valley of death where we would not be able to move from the academic discovery in a lab to a tangible, real clinical candidate.
We had been working with OCR on projects that were coming out of my lab that could have potential innovative avenues. My name's Anton Bennett. I'm a professor in the Department of Pharmacology. My lab focuses on the role of protein phosphorylation and cell signaling and how those pathways are connected to cell disease. It was clear that the Blavatnik Award was exactly what we really needed. We've taken all that we've learnt from the Blavatnik Award and we've identified some new targets, and we're going to use similar approaches on a completely different disease mechanism, utilizing some of that earlier information to guide us through a process where we can develop new therapeutics for this particular type of disease. The broad goal is that we can at least provide insight into new potential therapeutics to diseases such as liver disease that are going to help potentially millions of people.
OCR and Blavatnik support are really key at this very first step of moving from discovery down this long path. Without OCR's involvement, things will just be interesting discoveries, but they'll stop, they'll die. And so having a partnership from the Cooperative Research Office is what allows things to get down the road and to actually be tested and make a difference for people.